Hello, everybody. Welcome to an Applied Sciences webinar. Today, I am honored to introduce our speakers, Dr. Scheila Avila Silva and Gustavo Martinez from the University of the Caxias do Sol. Sorry if I don't pronounce them very correctly. Who will present their work published in SN Applied Sciences in this webinar. Before I give the floor to them, I just wanted to give a very short and brief introduction on SN Applied Sciences, which is an open access journal launched in 2019. And actually, it is a multidisciplinary platform for publishing the articles which reaches different fields of science, such as chemistry, material science, earth and environmental science, and engineering. So just a short, I will introduce the topic of this work, which is related to the work published in our journal. And in general, the research topic of the researchers coding bacterial genetic information into structural attributes. So I will give the floor to Dr. Silva, and you can just start. I will stop sharing my slides, so you can just share your slides.
So thank you for the invitation. Greetings for all. Thank you very much for taking part of this presentation, this webinar. So today, Gustavo and I will present the results that was published in the paper already presented. So we are talking about decoding the bacterial genetic information into structure attributes. So I am Professor Scheila de Avila Silva, and I am a professor and researcher at the University of Caxias do Sul in technology. Also, I am teaching, I have current teaching activities in the undergraduate level in computer science and biology, in biology also. And my main research is about the application of artificial intelligence in genomic data. So I have some experience in promoter prediction and genome assembling and annotation. And Gustavo is a PhD student. Please, Gustavo, can you present yourself?
Sure, thank you, Scheila. So I am Gustavo. I am a PhD student at University of Caxias do Sul. I have been working with Dr. Scheila for a long time. As her pupil, I try to continue her studies in delivering high quality genome annotation in microorganisms such as bacteria and archaea.
Okay. So so where are located in Brazil, because I think that the Caxias do Sul is not a big city and very know about. So we are in the south of Brazil and in a region of mountains. So we are around 800 meters above the sea level. And here in the mountain region, we have these points that are cities where the university have some locations. So the main campus is in Caxias do Sul, but we have another installations in cities around Caxias do Sul. In Caxias do Sul, we have a population estimated in a little more than half a million of inhabitants. So this is the picture of the city that we live. And the University of Caxias do Sul this is the map of the main campus in Caxias do Sul, the main location, and we have 85 undergraduate course, 18 master degree and 11 PhD degree course, and we have around 50,000 students in this year. So we have our website, and we will be happy to receive anyone that are taking this part of this presentation to have some visitation, or okay, we are very glad to receive you. So our agenda today is about some topics that we separate in gene expression. So we take a brief overview about how the genes have their products, so and the role of promoter sequence in the gene expression process, mainly in bacteria. After that, we will focus on the importance of promoter research, and then Gustavo will present the results of the paper about the study of structure and physical properties in promoter and bacterial promoter sequence. How genes make a product. So what is the main process related to this? So when we start to study the molecular biology, we know about the main dogma of the biology, so the central dogma. And the central dogma here, I put just two processes of the three main processes, because these two processes are related to gene expression. So we have the transcription. The transcription is the ability to DNA make a copy of the DNA sequence into mRNA sequence, and this copy in its form of mRNA goes to the ribosome, and in the ribosome this sequence can be translated in a protein. So this is a kind of a simple way to see how the genes make their products, but we know that there is a lot of steps between we have a lot of process that interfere in the synthesis of RNA and the traduction also. So there is a lot of checking points, regulating points in this process. And in the simplest way, it's okay, it's so easy, but it's not, because here, when we talk about the gene transcription, we have always the focus in the transcription, in the coding region. Now here in this picture, named the CDS. So the CDS, the gene itself, the CDS is, it's very important, but it's not only the CDS that have some importance in the gene expression, because we need to have some regulation points in the gene expression. For a bacteria have a short period of time, and the molecules in bacteria have a short lifetime also. So it's very important to be quick and response and have a appropriate response to environment change in order to survive. So in bacteria, the most of important regulations, gene expression regulations points are upstream the gene. So are upstream the nucleotide that we call TSS. The TSS is numbered as +1, and after that, here the TSS, and after that we have some coding region. The TSS is the first nucleotide transcripted but not translated. If forward the TSS, we have the TNSS. That is the first nucleotide that in the ribosome will be translated in some amino acids. So in this point, we have some structures that present some is a regulate point as the Scheila region. That is some location we help to the ribosome accumulation. And we have also the ITR and 5.UTR. So the ITR is a kind of region that make the release of it's a promoter escape region. And the UTR, it's a kind of region that make helps the strength of the not only the translation, but the transcription. But mainly in bacteria, it's very important not to regulate after the gene expression is not regulated after the transcription, because bacteria is a single cell living life. So there is its resource and its response need to be quickly. If need to be quickly, the bacteria cannot do a protein, do a total mRNA and just, okay, I don't need this product, so I can leave, I can destroy, and that's okay. No, because it's a kind of life that situations, if I have to give a response, okay, so it's not a bacteria, for bacteria is not able to take some, I don't know how to explain, but it's a kind of, okay, it's necessary to not think, but it's necessary to produce just the necessary. So the points that are regulation upstream the TSS are mainly the promoter sequence. The promoter sequence is the star of the regulation. Okay, it's not the only one. We have some transcription factors bind sites, but the promoter, it's a key regulator, because in this sequence, in this just 2 box of 6 nucleotides, the RNA polymerase binds and open the double strand of DNA and starts the transcription of the messenger RNA. So we're talking about the next slide. The next slide can show you how dynamic is bacterial life. So if there is some change in the environment, and okay, I need to survive of this high temperature or this osmotic pressure, so change dramatically. So the bacteria needs to survive. So the RNA polymerase binds in the promoter, open the double strand, and while the the mRNA are being synthesized, the ribosome comes and start the protein production. So it's a kind of very dynamic process. And so it's not possible for the bacteria make some unuseful product. The next, Gustavo, please. So what is the promoter sequence? Because we're talking about just a small piece of DNA that have so huge importance in promoter sequence, in gene regulation. And it's not a new study. It's a kind of, there is a history about promoter prediction. The studies of promoter prediction start with some two innovative papers that were published, just compilating and presenting some DNA sequence that was experimentally defined as promoter sequence. So first one in the first 80s, and this not second, but the last one in the 90s. So these were very innovative, and Lisser and Margalit published a compilation of almost 300 promoters, and we are talking about the early 90s, that the sequencing and genomic projects are not a reality. It's a kind of emerging technology. So they are so pioneers, and these two milestones in promoter prediction make some results that were impressive at the time. So these papers present for us that the promoters have some consensus sequence. So the region named -10, or the region named as -35 have some consensus DNA, so they are conserved into their DNA content. And of course, that if there is a pattern, if it's possible to see some nucleotide pattern, it's possible to do some statistics. So the next following, okay, we have the sequences. Following the sequences, we have the application of artificial intelligence in promoter sequence. Please, Gustavo. So the first papers about the application of artificial intelligence in promoter sequence was this one that taking some neural networks in order to predict if a given sequence could be a promoter or not. Okay, so this kind of, okay, it's easy. No, it's already done, and we have a good start of art in this subject. No, this is not a reality for us still. So we still have an open research objective when we talk about promoter prediction. Why this? So this is the picture of a promoter. So a promoter have the 35 element, a kind of consensus sequence as TT, G, A, C, A. Some spacer, some a length of non-conservative region of the promoter, and then -10, this TATAA10. So in biology, for the polymerase, the enzyme, the polymerase, it's not a big deal to change some nucleotides, or maybe have some -10 element being a short -10 element or a bigger -10 element. But in artificial intelligence, for the computational process, some change is very drastic, and it's not possible to predict just with the nucleotide content. So, Gustavo, please. So because the it's not so simple for bacteria. It has the bacteria need a so specific and a so quick response. We have a lot of proteins named sigma. The sigma factors, they bind in the RNAP, the polymerase, and give the specificity. So with some sigma 70, sigma S, sigma H, sigma F, every one of the sigma named as the gene that express them or their molecular weight. So this sigma confers for the polymerase the specificity that is necessary, because the genes that have a promoter recognized by sigma 7, it's about the function of our housekeeping genes. And if you are talking some stress or some osmotic or shock heat shock response, we're talking about other sigma factors. Escherichia coli have some 6 or 7 sigma factors known at this moment, but we have bacteria with 20 or more than 60 sigma factors. So it's not only the location or the function of the gene that is related for this specificity. Each sigma each promoter sigma recognized by a different sigma factor present a different consensus sequence. Please, Gustavo. Yes. So if we take the main promoter sigma, that was the sigma 7 here, we have the main and general consensus, TTG, the GACA region, and the pyrimidine box region, TAT. But if we are talking about the sigma 28, we are talking of another region, another consensus region, so another nucleotide content. So we can't use the same approach for all promoters, because all promoters are different. So all are promoters, and the polymerase recognize them as promoter, but they are not the same. If they are not the same, the artificial intelligence it's not able to understand some little modifications in the nucleotide content. So why the promoter prediction is an open object yet? Because we have a lot of studies that need understand the basic science related to promoter sequence. First of all, we can use the promoter prediction, the promoter study, to understand the evolution of the gene regulatory network. So we can make some comparisons and conclude what is the same, what is different, and what we can make some evolutions, thoughts and hypotheses about how the gene expression regulator was evolving in among the taxons and the organisms. Another good, and I think that's very important and very how can I say, it's very important and present some good results, is how to understand the relationship between the parasite and the hospitator. So how can we understand the pathogenesis? So this paper presents some insights about how we can prevent some bad behavior of bacteria. They put in a bacteria some false 54 sigma. So into the bacteria was a lot of true and false sigma 55 sigmas molecules. So the bacteria was not able to carry out all the pathogenic. So the expression of the genes recognized of sigma 54, sigma 40, 54 promoter sequence was not able to do. This is a kind of a good hypothesis about new medicines, new chemicals, new products for this relationship among us and our pathogens or another organisms. Please, Gustavo. And the last one, we need to take in account the genetic engineering, because we have good techniques, but we need to remember that the gene just will be expressed if the promoter is recognized as a promoter. If the promoter is not recognized as a promoter, there is no gene expression. Doesn't matter what is the content of the gene, or if there is some mutation or not, because we need the first step is the promoter recognition. So this a promoter that is recognized across the organism is very important to genetic engineering and other techniques related. So taking this into account, what we think, okay, the sequence for the promoter is very important. Okay, we have this knowledge, but the polymerase is not taking just this knowledge when it bounds in the DNA. So what is the structure and physical properties about the promoter sequences in bacteria, taking Escherichia coli as an example? So Gustavo, please.
Okay, so thank you, Scheila. I will continue from now on presenting the results that we have obtained with the study, namely DNA structure and physical properties, review peculiarities in promoter sequences of the bacterium Escherichia coli K12. So the main premise that has conducted us in developing this study is by taking a look at a DNA sequence, not only by the 4 letters that compose it, but due to a series of physical attributes and structure attributes that can be used in order to represent a DNA molecule better than just sheer numbers. If we consider the DNA alphabet, it's quite limited. It has only 4 symbols. So whenever we're working with mainly artificial intelligence techniques, we need to find ways to represent these symbols, these letters, as numbers, for example, in which we have an infinite alphabet and many combinations might be done. So we have selected here 3 different parameters in which we have coded genetic information into. All of these parameters, they are totally associated to the sequence itself. We're going to explore these a little bit later, but for example, the levels of stability in a given DNA sequence tends to be different when we compare coding regions and other regulatory regions, such as the promoters. So it's by taking a look at this conservation that is found only in the promoters that we try to code it into numbers. The 3 features that we have selected to do so are stability. We have also enthalpy, which is a thermodynamic feature. And finally, we also have DNA stacking values. All of these are good representatives of genetic information, and there has been reports of these being used as distinguishers, and they are able to produce genetic variants such as promoter, non-promoter, and other regulators as well. So by using this codification, we could get to 3 different segments of results. The first one is when we can totally represent a DNA sequence by taking a look at its profile and say exactly which are the areas where transcription factor proteins, in that case, since we're talking about bacteria, the sigma factors, they bind specifically to that. There should be a difference in the stability, in the enthalpy, and in stacking levels in that particular region, because it's the region that the transcription factor proteins, such as the sigma factors, they bind specifically to those DNA areas. Furthermore, we can also take a look at how these 3 features are correlated, if there is one that can better represent a given sequence, a given sigma, and so on. And finally, the main reason for this is for us to use this DNA codification in an attempt to find promoters in unannotated genomes. The methods and data sets that we have used to do so, we have selected here a selection of promoter sequences that are listed in the database RegulonDB. So we have here a number of sigma factors in different groups. And we have here in our second column, the total number of sequence for sigma family. Also, there is some difference in the number of associated genes to that. It tends to be lower since we're talking densely packed genomes. And as Dr. Scheila was mentioning before, we need to consider that these organisms, they have a very limited lifespan, and also their supplies need to be totally optimized in order for them to thrive in wherever environment they are into. All of the sequences that we extracted from RegulonDB, they contain 81 nucleotides. This region has been reported as containing the core of the promoter. So we are not missing anything by comparing only with 81 nucleotides. And furthermore, we have here the AT percentage, which tends to be a little bit different between the sigma factor that we have. Moreover, we have selected other sigma 24 promoter sequences from other bacteria, not only E. coli, as an attempt to validate our findings. And we have also delivered a 2-level form of control sequence for us to compare the real ones versus the controls. These control sequences encompass a shuffled version of the original sequence, and we have also selected the immediate downstream sequence, which is going to be transcribed into an mRNA molecule and finally into a functional protein to try to promote that gene variation. The promoter is different from the coding region. We have here our features, the parameters that we have chosen to codify genetic information into. So we extracted these from the studies that have already performed these calculations. And according to each duplex, each set of 2 nucleotides, we should have a different value of enthalpy, stability, and base pair stacking, as this table is presenting to you. We have a database with more than 100 features, more than 100 structural and energetic parameters that we can use to convert genetic information into numbers. All the tests that we have done here, they were done following the R environment for statistical programming. And we have also normalized our data, since each one of the features, the parameters that we have to codify genetic information, is found in a different scale. So we decided to bring all of these together into the same scale that was 1 and 0 normalization. And lastly, we have also employed sequence logo profiles through the web logo tool to analyze particularities in the sequence level of the sigma families that we are testing. When we first start exploring our results, this is when we get the sigma 24 promoter sequences, which according to this image that we have here, is not showing good patterns to us. As we can see, each one of the lines is representing one of the selected parameters, but as a human eye gazes upon this image, there is no much information that we can extract about. So the first thing that we need to discuss about it is how the sigma 24 promoters are listed into the website RegulonDB, because around 80% of these promoters, they have not been experimentally identified. They have only been classified as promoters by computer techniques, by specifically machine learning algorithms. So when we are dealing with data that hasn't been curated, we can see that there is not much valuable information we can extract out of these. And then we can totally relate these with the need of experimental biology. Both Dr. Scheila and I, we work with bioinformatics, and a lot of times people say that we are going to replace the experimental biologists in the future, but that's not the case. In these, a computer technique couldn't extract much information because the data hasn't been curated yet. So there's still a need for experimental biology. We can still coexist, and experimental biologists using data from bioinformaticians and vice versa. When we move to other sigma factors, we can see that the figure is slightly different. Let's focus here, for instance, in sigma 28. This peak here, it matches the binding site of transcription factor proteins, in that case, the sigma factor, -10. And the -10 is visible in all of the families that we have here. There is a peak in our physical features that is telling us where the sigma protein binds the DNA. And not only that, but we can see that our three chosen features, enthalpy, stability, and stacking, they show a similar level of behavior. As you can see, the lines are somehow overlapped. And then we have tested for correlation in how these features are correlated. So we have found that enthalpy and stability, they are moderately correlated, presenting a P of 0.53, and then stacking and stability have a median level of correlation of 0.47. So the three features that we have chosen to explain genetic information are showing a similar behavior. So this is telling us the similar pattern that we can extract from these three features. We're going to see later that they behave differently. They are formed upon different bases, but we can see that their behavior is quite similar. Moreover, we have been able to identify the binding site of transcription factors, in that case, the sigma. I have selected here these images of sigma 28, the upper one, and the bottom one, we have sigma 54, I'm sorry, in which if we take a look at the line that we have created for the features themselves, and we overlay these with sequence logo profiles, the letters of conservation, the conserved nucleotides here, we can see that in the spots that we have more conservation, it's also the spot that we have our peaks identified by the images. So we can use the physics, the physical profiles to explain the consensuses, the preferences for certain nucleotides, this big island here of AT, and we can also use the nucleotide information to explain the physics. So one is assisting each other. Moreover, when we follow the same rationale to test other organisms with their sigma 54 promoters, we need to consider that there's not much, there's no such studies on these organisms as we would have with E. coli, which is the most studied organism, and talking about bacteria. But we have found similar patterns as well in both the places where the sigma factors of sigma 24 should bind the DNA, which is -12 and -24. This confirms that these rationale might be transferred to other organisms, and we don't need to hinge on E. coli only. When we take a look at a comparison with control sequences, the two levels of control, we can see that there is a very good possibility of distinguishing promoters and non-promoters independently from the sigma factor that we are comparing. For example, in all of the highlighted cells here in our table, we can see a high significance level found between the promoters of that given sigma and its two control levels. Even the ones that are not highlighted, they presented an interesting level of correlation, of significance between the promoters and between the controls. So that's a good way for us to start designing a predictive tool that tries to identify promoter sequences based on their physical attributes. When we take a look at each one of our features separately, we first here start with enthalpy, in which this feature itself, it's used to describe the level of thermal stability that is found in a DNA molecule. And there are several processes that happen in a cell that might affect the temperature, finally the thermal stability of that, such as heat exchange, the storing of genetic information, the mass transport inside and outside of the cell. All of these processes have been reported as contributors to changing the temperature of a cell. So that's how enthalpy might be used as a way for us to explain promoters. If we take a look here at the different sigma families that we have chosen to enable here in this image, we can see that when we compare the two forms of control, the coding and the shuffle, the promoter is always in the middle. There is a difference between these two profiles. So we can explain promoter activity by converting the promoter sequences into enthalpy. It should be different from the two forms of control that we have selected to do so. When we go to our other features, we have here DNA stability. So the promoter sequence tends to be a region with lower stability because the RNAP is constantly binding to this region. And for this to happen, we have the need of a balanced structure. This balanced structure is generally found because we have differences in the hydrogen bonds between ATs and between CGs, the nucleotides. And the promoter sequence tends to have different ATs and CGs. So with this difference that we have between the promoter, we can basically explain the stability level of it that tends to be lower. For instance, we have ATs with 2 hydrogen bonds and we have CGs with 3 hydrogen bonds. So by taking a look at the images again, we highlight here to sigma 28, in which the promoter is totally different from the 2 forms of control. In the other sigma families, this is not that clear, this is not that visible, but we still can locate here the transcription factor for binding, the transcription factor protein binding sites. And finally, when we analyze DNA stacking, we have here the vertical relationship between our base pairs. It's basically a similar pattern of what we would have found with stability, but not horizontally speaking. It's vertically because the DNA is all tangled, and for this process to happen, we need to have stacking forces between the base pairs. And some early reports have found that the difference between binding strength on stacked ATs and GCs is different again. So it makes sense for the promoter to have different stacking forces because it's majorly composed by ATs instead of GCs. The same thing that we have explored with stability. And then highlights again to sigma 28, in which the promoters are very off from the two forms of control that we have here. So this tells us that we can use these three features separately and all combine it in an attempt to explain the inerts that happen with the promoter region itself. Finally, we have come up to the conclusions. So we have found that the three features that we have tested here are good representatives of promoter activity. Promoter sequences they could have been explained by a combination of these three features. And then we also create the potentiality for using this conversion into physical attributes in an attempt to classify promoters, talking about promoter prediction, talking about computer techniques that are that get a sequence as an input and try to say if that given sequence is a promoter or not. Based on what we have collected here, we can say that a study such as this might be attempted. So that's it. Thank you very much for your time. We are now open for questions.
Thank you for your presentation. It was very detailed. So if somebody has questions, you can just type it in the chat box. But I have actually two questions. You mentioned that it's not possible to replace computer science or computer techniques by biology. So actually you always have to go to the laboratory and do the really biological experiments. Can you like name some of the challenges? So I imagine you think that there are some errors in these computational techniques or methods. Can you a little discuss that?
Yes, sure.
Yes,
you want to start, Scheila, please?
Yes, I can start. So this is a kind of good question because when we talk about statistics or artificial intelligence applied in biological data, it's always there is some degree of mistake because artificial intelligence always have some degree of mistakes. It's not different in biology. For instance, there is several tools that make the promoter prediction. And if you have the same sequence put as input data in 3 or 4 bacterial promoter prediction sequence, the result's not the same. So it's not that the in silico or the computational it's not good enough. No, it's very good, but it's good to lead for a more assertive or a less expensive, even in money or time experiments. So it's very important to put together the in silico and the in vitro or in vivo experiments. So we need to carry out 2 or 3 different computational approaches to recognize some sequence of even genome annotation, promoter prediction, transcription factors, binding sites prediction, and other, because it's in computations, it's a prediction. Even in the experimental, we have some degree of uncertainty. So
Okay.
It's
So they
yes.
always stay like as complementary techniques together. You always need both of them. And like you have an overview of like in which direction this artificial intelligence is going on, like in which fields it's more helpful. So can you just also comment on that? Let's say if regarding in your field, which specific subdisciplines can be more influenced by artificial intelligence, or let's say in future, what breakthrough achievement you are going to expect that happen, and it is because of using artificial intelligence in your field?
Okay, Gustavo, do you want to start?
Yeah, sure. This is actually a very good question. So, for example, I am extensively working now with archaea. So those are other forms of microorganisms, totally different from what we have presented here about bacteria. And the interesting thing about archaea is that these organisms have been classified as the third domain not so long ago, like 40, 50 years. Talking about science, it's not a lot of time. What happens is that archaea are organisms that live in every kind of environment that we can think of. We have the extremophile organisms. We have organisms that are not easy to cultivate on a lab. So right now we're conducting a study that uses artificial intelligence in order to promote genome annotation on archaea. Since it's nearly impossible for you to cultivate some of these organisms into a lab, the artificial intelligence is doing a good job. We can annotate that and we can understand more about the regulation of that organism by taking a look at bioinformatics itself. Is that enough to promote conclusive studies about these particular organisms? Probably not. Probably we need to coexist with experimental biology. But what happens when experimental biology in this particular question is not available for now? Because come on, we're talking about organisms that live and thrive upon 100 degrees of temperature. It's not easy to do in a lab. So by doing that, we can create some demands that experimental biologists are going to test on, are going to be able to validate. And the two questions that you have asked us here, they are kind of entwined and connected. So for instance, when we got our study here that we presented, we saw that for Sigma 24, our method couldn't be applied because the sequences that were given to us, they were not curated, they were not validated. So for that particular case, we would need experimental biologists. But at the same time, the experimental biologists for conducting other studies, they would need us. They would need the curation curation by bioinformatic techniques. So it's not trying to place these into hierarchies. For example, we first have experimental biology, the most important one, and then we have bioinformatics. No, depending on the demand, one is going to assist the other and vice versa. And that's, in my opinion, what is beautiful about it.
Okay, perfect. Thanks. I mean, we are run out of time more or less. We have just 4 minutes. So my very last question. Well, I mean, let's say your research is definitely in the field of biology. So if you want to bridge it to other fields of natural sciences, so if you have to choose between chemistry, material science, and probably engineering, which of these like you will find more connection between these fields and your field? So if you want to describe your field as multidisciplinary, which of these you will choose? Chemistry, material, or engineering?
I think chemistry, Gustavo, because one of the goals of the future about the promoted prediction is the future of the medicine. Understand how the bacteria make our sick, make the living other living beings sick. So this is the future, how this partnership, how we can put together our knowledge with the chemistry of the compounds knowledge.
Okay, thank you. Thanks again. It was very interesting and very interesting talk and topic. And yeah, thanks for your devoted time and thanks from the participants. So I have just recorded this webinar and we will share it again with you and participants, and also we will do some marketing campaigns on that. And please don't hesitate to contact me if you had any questions. Thank you.
Okay. Thank you. Thank you.
Thank you. Have a nice day.
Thank you, everyone.
Bye.
Thank you. Bye.